Yep. Yep. So, how we study cells, we use microscopes, right? We looked at light microscopes, now electron microscopes. We'd already looked at scanning electron microscopy. Now we're going to look at transmission electron microscopy. And we sometimes abbreviate that to TEM, and the images that they produce to TEMs, transmission electron micrographs. Now here's the way they work. Is you take your specimen, you have to embed it in a plastic resin, and there's a whole series of steps you go through, and you stain it with heavy metals. Osmium tetroxide is an example of one, but you stain it with heavy metals. Different parts of the cell absorb the stain differently depending on their chemical composition. Then you use something called a microtome to take very, very, very thin slices. Okay? And it's those very, very thin slices that you then put in your electron microscope, you bombard it with electron beams, and the electron beams pass through. But they don't pass through equally, right? Because some parts are stained with a heavy metal. So the parts that are heavily stained, the electrons don't pass through so well. And there's a sensor on the other side that's collecting electrons that are able to pass through. So if a lot of electrons pass through, it looks kind of white or light grey in colour. And if only a few electrons pass through, then it gets progressively darker shades of grey to black. So that's how you're able to generate an image with an electron microscope from those thin sections. It's kind of similar to light in a way, isn't it? When you use a light microscope, but light is coming up from the bottom, yeah. And areas that let a lot of light through look light in colour. Areas that don't let a lot of light through, because they're presumably because they're dense, then they don't let much light. Then they look dark. So we use TEMs to study the internal structure of cells very often. Remember, you've got fantastic resolution with an electron microscope, and you can look at these structures within the cell. So this is an example of a TEM. In fact, it's a TEM of an SEM that I showed you last time, the last SEM, of the cells that line your trachea. All right? So here you can see a section through them. Very, very thin, and it's in 2 dimensions, not 3 dimensions. Is everybody okay with that? Can you sort of relate that to the one that I showed last time? Yeah. Okay. Typically, any dark line that you see like this, or any dark line that surrounds something is membrane. It might not be the cell membrane, but it's membrane. In this case, that's the cell membrane. But all of these are little internal structures bounded by membrane. Here's a TEM of a mitochondrion. All right? And those dark lines, again, are membrane. So there's membrane, there's membrane. Some of these other things are other molecules. In fact, I think these are ribosomes. But there's a membrane, and there's an inner membrane which forms all these folds. These, do you remember we tried to look at these in lab, but they were far too small to see under the light microscope. We couldn't even see them, could we? But look how much detail you can see under the electron microscope. Why do we have folding? What's the folding for? Increased surface area, yep. All right, so cell fractionation is another technique that we use to study cells, and that's exactly what we did in lab. So I'm going to really fly over this since we've already done it. We did it in lab, yep. So it should just be a very quick revision. It's where we separate the organelles to study the function of each of them. First, we disrupt the cells, we homogenize the tissue. We can filter it as well to get a filtrate. Then we'd centrifuge it. And we centrifuge it progressively faster and longer to get the different organelles in different fractions. We use shape, density, size as the physical parameters to separate them. And these are some of the terms that we looked at in lab, right? The homogenate, centrifugation, the supernatant, the pellet. Good. Should just be a revision. Yep, on that one. All right. And there's a little diagram just showing those sort of sequential steps that we go through. Homogenize it. Filtration is not shown in this one. Sometimes we need to filter it, sometimes we don't. Depends on the tissue that you've homogenized. If it's a soft tissue like liver, then usually you don't need to filter it if you completely homogenize it. All right. So concept number 3 then is we're going to look at this dichotomy between prokaryotes and eukaryotes, the different cell types. every organism on planet Earth is composed of one of two cell types. You are either a prokaryote and made of prokaryotic cells, or a eukaryote and made of eukaryotic cells. Now, the word prokaryote literally means before nucleus. Pro means before, karyon means nucleus. The word eukaryote literally means true nucleus. Eu means true, karyon refers to nucleus, true nucleus. So you're either made of one, an individual is either one, or many cells. All right? You happen to be multicelled, made of many cells, and your cell type is eukaryote. You're made of many eukaryotic cells. A bacterium, on the other hand, is unicellular, one cell, and they are prokaryotic cells. So the prokaryotes then encompass the two domains, bacteria and archaea. The other domain is what? Anybody remember what the third domain is?
Is it eukarya?
Eukarya, good. And we can break down domains further into what? Kingdoms, good. So what are the four kingdoms of eukaryotes? I'm pretty sure I heard them all. Good. I'm pretty sure I heard them all. I heard them all somewhere in there. Plants, fungi, protist, animals. Good, good job. All right. Okay, so is everybody okay with what kind of organisms are which cell type? Okay. All right, good. So let's have a look a bit deeper at these cell types then. All cells, regardless of whether you're a eukaryote or a prokaryote. This is all cells. Can you think of any features that all cells have?
Cell membrane.
Good, cell membrane. What else?
Yeah, probably mitochondria. Is that a feature, or is that just...
It's not a feature of all cells.
Okay.
All cells do have... Oh, what was the one you just said?
DNA.
The one before that?
Membrane.
Cell membrane.
Cell membrane. All cells have a cell membrane.
But not all have a cell.
And what was the next one you said?
DNA.
DNA. All cells have DNA. Good. Is there any other feature of all cells? So they've all got a plasma membrane, cell membrane. They've all got this sort of semi-fluid, almost jelly gel, runny gel-like substance called cytosol, which forms the cytoplasm. All cells have that.
So if
All-
someone says cytosol versus cytoplasm, it's the same thing?
You know,
Because I thought there was a difference, but I'm not.
there is a difference.
Okay.
And honestly, I use them interchangeably. I probably shouldn't, all right? But I'm probably going to. I think cytoplasm is a little more old-fashioned, or it might be a little more on the British side. But I think there is a distinction between them. I'll look that one up and tell you. If there is, it's a subtle one, I think. All right, and all cells have DNA. All right, and the DNA is arranged into chromosomes, and that's where you find the genes. Good. All cells have these structures called ribosomes, and we'll talk a lot more about ribosomes in a moment. And ribosomes, they kind of make proteins. They're the site where proteins are assembled from what? Amino acids. Good. Yeah, good. All right. So that's kind of the assembly site. All right. So prokaryotes then. Let's look at features that are specific and unique to prokaryotes. Firstly, they have no nucleus. The DNA is not contained within a nucleus. There is no nucleus. When you look at a prokaryotic cell, you can't see a nucleus inside the cell. There isn't one. They do have DNA though, and the DNA is in this region of the cell that's called the nucleoid. And it's just a region of the cell where the DNA resides. In other words, the DNA is not strung evenly throughout the cell. It's sort of concentrated in this little area called a nucleoid, which happens to be dense, stain more densely on an electron micrograph. And there are no membrane-bound cell organelles. So they don't have mitochondria. They don't have chloroplasts. All right? There are no membrane-enclosed cell organelles. They do have membranous structures inside the cells, but they're not enclosed within membrane. Okay. So the eukaryotes. Oh, sorry. So the cytoplasm. See, I've used that as compared to cytosol. I'm really not sure if they're completely interchangeable, but I will look that up. So the cytoplasm is enclosed by a plasma membrane. Now for bacteria, what do we usually find just outside of the cell membrane?
The cell wall.
Cell wall, and sometimes they have an extra part called a capsule. Yep. Good. All right, so in eukaryotes then, the DNA is enclosed with a nucleus. Eukaryotes have a nucleus, a membrane enclosed organelle where the DNA resides. Okay. Eukaryotes do have membrane enclosed cell organelles. All eukaryotes have mitochondria. Did you have a question?
DNA is in the nucleus.
Of eukaryotes. Okay. Some eukaryotes have got a chloroplast, not all of them. Animals and fungi don't. Plants and many protists do. And eukaryotic cells tend to be larger than prokaryotic cells, by and large. The protists and the plants tend to have pretty big cells. The animals tend to have a little bit smaller cells, and the fungi tend to be a little bit smaller still, by and large. All right, is everybody okay then with the sort of similarities and the differences between eukaryotes and prokaryotes, yeah? All right, so here are some pictures then. One of them is a diagram of a bacterial cell, sort of a typical rod-shaped bacterium, and there's a transmission electron micrograph, and the different structures are labelled. So there's the nucleoid, that region which is not enclosed within a membrane, the region where you're going to find the DNA. Okay, you do have a cell membrane, which on the TEM looks like a very thin dark line. This dark structure outside the membrane is a cell wall, and this bacterium has a capsule as well, which is like an extra added thick part to the cell wall. Some bacteria have flagella. Right, these whip-like structures which are able to whip around and enable the organism to swim. Some bacteria have these fimbriae which enable them to kind of attach to surfaces or other things.
So capsule you just find on bacteria?
So a capsule, no, not all bacteria have a capsule, but some do. Oh. And you can think about it as an extra part to the cell wall. Oh. All right, an extended part of the cell wall. It's exterior to the cell membrane. Now, to contrast that with a typical eukaryotic cell, so this is a sort of diagram of, I don't think there's any eukaryotic animal cell that actually looks like this, but it's a diagram of one showing all the different parts in a eukaryotic animal cell. Please note that there's lots and lots of compartments, all separated by membrane. Okay? So we're going to look at the function of most of those structures in a moment. Now contrast this cell to this one, which is a plant cell, also a eukaryote. Again, similarities, but certain differences. I want you to have a look at the cell membrane, just there, which is sort of yellow in this diagram, and the cell wall is exterior to it. Okay? Think about everything inside the cell membrane is where you find the cytoplasm and all of the organelles. Okay? Good? So we're going to look deeper at these. We're going to focus on the animal cell, the eukaryotic animal cell, and look at all the different subcellular components, what they do, and how they're structured. All right. So concept number 4 then is I want to look at the size limits of cells. Why are cells the sizes that they are? Now, what was the biggest cell I showed you? ostrich egg, right. Now that's atypical. A bird's egg is really an atypical cell, right? I mean, they're huge, obviously, but they're kind of atypical. So your average animal or plant cell is much smaller than a bird's egg, all right? So cells can only be so small and they can only be so large. So when you think about large cells, try not to think about the bird's eggs, even though technically they are. You've got some very long cells in your body that happen to be extremely thin. You've got a neuron, right, that runs from the base of your spine to the end of your toe. That's a very long cell, isn't it? You've got a muscle cell, right, just here, that runs from this entire length, okay, well over 30 centimeters long. But they tend to be very, very thin. So many cells have a size limit. They can't be bigger and they can't be smaller. So let's have a look at that. What do you think limits how small a cell can be?
Its function. Organelles.
What about if they don't have organelles? Or at least membrane-enclosed organelles? What's the smallest cell that we looked at that I shared with you? A mycoplasma. Teeny, 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 tiny. It's a kind of bacterium. It's prokaryote. Very, 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 very small. That's the smallest cell we got. Why can't we get smaller than that? Stuff won't fit. You need a certain amount of stuff in order for the cell to function, right? Once you get so small, you can't do without things, right? It's a bit like when you travel. Some people travel really light. I have a friend that I travel with sometimes. Oh my goodness, he goes on these international trips, sometimes for weeks, with a bag about this big and about that wide. That's it. All of his gear. It's unbelievable. I took a lot of, I thought I was good when I traveled, but this guy was amazing. But he still needs a bag a certain size. I reckon that's as small as you can get, because you need a certain number of things, right? Are underwear optional?
Yes.
When you travel, they actually kind of are, but I mean, let's just say that they're not. All right, let's just say that they're not. So you need to pack a certain amount of stuff in there. Yeah.
So what do you do with like souvenirs and stuff? Where would you put that at?
So it's kind of different when you travel back. All right. Sometimes what I'll do, like when I go to England, I actually bring a whole other bag in my checked bag, because I bring back a load of food things that you can't buy here, that are not easy to buy here. So I usually pack out another bag with things. So I think it's okay on the return journey to bring another bag. Right? That's my personal opinion. But don't bring much stuff. You just got to cart it around, yeah? You get really good at ditching things after you've had to lug a big bag around. All right, so you can only get so small because you need a certain amount of stuff to carry out all the basic essential functions. Okay? All right. But what limits how big a cell can get then? What limits the maximum size of a cell? Again, don't look at the exceptions like the bird's eggs, your average cell. So let's think about that. Why can't it be bigger? Because you think bigger, we can get loads more stuff in there. Why are you made of lots of different cells, not just a few big ones? All right? There's a lot of answers to that question. But think about the metabolic requirements of a cell. They kind of set the upper limits on the cell size. So if here's a cell, we've got a certain amount of stuff that we've got to fit in there. And you've seen pictures of the kind of things that are crammed in there, right? What kind of things have got to go into the cell? Let's name a couple of them.
DNA.
Well, DNA's already in there, so let's think about things that there's a constant traffic into and out of a cell. Constant flow of things in and out. What things are constantly flowing in?
Water.
So water is. There's a movement of water. Yeah. Water's not really used up in the cell. I mean, it is a little bit. We talked about hydrolysis reactions and we talked about dehydration reactions producing it. Yeah. Proteins do move in and out. So amino acids. How about we just say monomers? Monomers for macromolecules need to continually move in. Somebody said proteins, yeah? Well, let's think about macromolecules need to move out. Now, cells also take in macromolecules and cells also take in monomers. So I guess let's put some two-way arrows to that. And water actually moves out as well. So let's do two-way arrows to that. What about substance which moves in but doesn't really leave in its original form?
Oh, at first I thought you were huffing and like disappointed.
Oxygen, good. Oxygen, one pretty much moves into cells that use oxygen and it leaves. CO2 leaves. Okay. What's another substance that enters but doesn't really leave as itself?
Would you say like sugars?
Definitely, I would say sugars. For example,
Glycogen or glucose. Yeah, that's what I meant.
glucose moves in. Now, of course, some cells take in glucose. They store it as glycogen and glucose leaves as well. But sometimes, very often, this glucose is burned, disassembled, right?
Yeah, and the leaves it's like.
And it leaves, the carbon leaves in the form of carbon dioxide. And I know I've got water leaving, but I'm going to add it over here because we've got glucose and oxygen, carbon dioxide and water. That's cellular respiration. Okay? For an animal cell, at least. If you're a plant cell,
Right, the opposite.
it's kind of the opposite, isn't it? These are going to enter a lot of it, a lot of carbon dioxide, a lot of water, and oxygen and glucose are going to be exported. So it does depend on the cell. But the point is, there's an awful lot of traffic into and out of cells, an awful lot. Okay? All right, it's like a very, very busy factory. Stuff's coming, stuff's going really quickly. Now, the bigger the cell, the more stuff it needs, right? Just like this, the bigger the factory, the more things that are going on. So, there's an awful lot more traffic going into and out of a big cell. All right? Now, if you can think about it almost like a factory, and then we've got doors there, and a door there, and a door there, and a door there, and a door there. Here's what limits how much stuff can get in. How many doors we've got, right? If you don't have enough doors, and you make this bigger, you're going to be starved for something. Something's not going to be able to get in because there aren't enough doors to let it in quickly enough. Is everybody okay with that analogy? So as a factory gets bigger, things go in and out faster. It can only get so big before, that's it, no more doors. All right, not enough outside area to allow the movement of substances in and out quickly enough. All right, so we call this, the sort of space where you've got the doors, that's its surface area, the area of its surface. And the space inside, we're going to call its volume. So we're going to look at this in terms of the surface area to volume ratio. As a cell increases in size, its volume grows proportionately much faster, much greater than does its surface area. It's not a one-to-one ratio. So we can add more volume, but we don't increase the surface area at the same rate. So we end up having big volumes, lower ratio of surface area, and that doesn't give us enough surface area to get things in and out. So here's the diagram that's in your book, and I'll sort of go through it column by column to illustrate this. So let's just think about this as a small cell and this as a large cell. Okay? Now, cells aren't square, not many of them anyway, none in your body. So let's just say that this cell is 1 centimetre by 1 centimetre by 1 centimetre. All right? If you do the math, the surface area of that cell, each face is 1 square centimetre, right? Is everybody okay with that? And it's got 6 faces, 3 that are visible, 3 that are hidden. Yeah. So its area, its surface area, is 6 square centimetres. All right, now let's look at this cell. This cell is bigger, and it's 5 centimeters by 5 centimeters by 5 centimeters. No cell is that big, but I'm using these because it's tangible, isn't it? 5 centimeters. So, what's the surface area of this one face? 5 times 5, 25. So each face is 25 square centimeters. Yep. It's got 6 faces. So that's going to give it a surface area of 150 square centimeters. All right. Now let's have a look at its volume. This little square, right? It's got a volume of 1 times 1 times 1. 1 cubic centimeter. 1 cubic centimeter. Everybody okay with that? Real simple. So the surface area to volume ratio of this little one is 1 to 6. Sorry, or 6 to 1 it should be. If we're looking at surface area to volume ratio. All right, 6 to 1. Now, of the big one, let's calculate its total volume. It's going to be 5 times 5 times 5. 5 5's 25. 5 25's are 125. The surface area to volume ratio now is 1 to 1.2, or 1.2 to 1 if we were looking at the surface area to its volume ratio. This is much, much less, isn't it? 6 to 1 versus 1.2 to 1. So that shows you that as you increase in size, your volume gets much bigger in proportion to your surface area. Now, so rather than have fewer very large cells, we break things up into very small cells. We could take this exact same volume, right? If you broke it up into little cells, now you're back down to that 6 to 1 surface area to volume ratio. Good? Okay. There's one other thing I want you to think about. If here's the cell, here's a nice big cell, and here's a nice small cell. Okay? I want you to think about the distance from the middle to the outside. Middle to the outside. When substances enter a cell, now they can be transported around, but many of them get to where they need to go by diffusion. All right, they rely on diffusion. And diffusion is a slow process. We'll talk about diffusion properly at a later time. But diffusion is a slow process. They need to diffuse this distance. Now, diffusion works great, it's fast enough over small distances, but not big distances. This cell, nice and small, diffusion, things get to the center fairly quickly by diffusion. The big cell, they don't get to the center big enough by diffusion. Okay? All right. Is everybody okay then with that surface area to volume ratio? And it explains why cells can't get too big. Yeah? So, relatively inactive organisms that don't have high metabolic needs, their cells generally tend to be a little bit bigger. They can get by with that. If you're very metabolically active, maybe you can get by with having smaller cells. Now that's sort of very general, all right, but it illustrates the concept a little bit. All right, so at some point then, the demands of the cell for things like oxygen exceed the rate at which they can move into the cell because the surface area is just too small compared to its volume. Okay. Now I know I've drawn these squares in 2 dimensions, but we're talking about volume. All right. All right. Everybody okay with that concept? Okay, good. All right. So now we're going to move on to concept number 5. Now we are going to focus on animal cells and their eukaryotes. I will mention some plant cells. I don't think I'll mention fungal cells very often. I might mention protists, but we're primarily going to look at animal cells, and we're going to focus on the eukaryotic cell. All right. So do you remember that first video I showed you? Fantastic animation with the great music. Yeah. All right. So I want to show you another one. It's not quite as is animated in a little different way, but I think it shows you the cell in a slightly different but an equally good way. So this is a tour of a plant cell, all right?
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. All plant cells have mitochondria, just like animal cells do. Sugars produced by photosynthesis are broken down and converted to ATP in mitochondria. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
Right, now contrast that a little bit with an animal cell.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Good?
Holy poop.
All right. So we're going to break that down and look at it bit by bit. And I promise, so I won't play this video again, but by the time we've reached the end of this chapter, you'll go back to that video and just say, "Phoop, got it." Okay. All right. So eukaryotic cells then. Eukaryotic cell has got lots of internal membranes. And that partitions the cell internally, forming different spaces and different cell organelles. All right? So lots and lots and lots of compartmentalization by membrane to make different spaces, different compartments, and then cell organelles. Give me an example of two cell organelles in an animal cell. Good. Mitochondria and nucleus. Good. All right. So compartmentalization is an important feature of eukaryotic cells. When we break things into their compartments, we separate processes and reactions. You can almost think about it like your kitchen. You've got an oven to cook in, freezer to keep things frozen in, a microwave maybe to heat things up in. It allows for movement and storage. So substances can be moved around the cell without interfering with other processes, and you can store things in those different compartments. Some things are needed quickly, some things can be stored for long-term storage. And it enables you to create different localized environments. Maybe some environments have a different pH to their surroundings. Some little environments might have different ion concentrations to their surroundings. So plant and animal cells have most of the same organelles. They're very, very similar in the organelles they've got. There are differences, but by and large they share the same organelles. There's one very key difference. What organelle do plants have that animals don't? Chloroplasts. Good. There are some others, but... All right. So, now we're going to take a good close look at all the different components that make up a cell. Now, here's what I want you to do. I want you to take a clean page, a fresh piece of paper, all right, and we're going to draw a cell bit by bit and add to it as we talk about the different organelles. Okay? Now, my goal is that once we've constructed the cell, that you'll be able to draw it and label it within 2 minutes. That's the goal. Now, 2 minutes. I don't know, how long are you stuck at traffic lights for?
Oh, sorry.
Far too long. I reckon 4 traffic lights and you should be able to, worth of time, you can draw a cell and label it. It's no time at all. So, do this often, right? Draw the cell and label it often. And I've got to tell you, when it comes to that exam after spring break, you'll ace it, no problem. Draw it, label it, and then annotate it. Okay? How many of you got little brothers and sisters, little kids? Babysit anybody? All right. Sidewalk chalk. Or you would say sidewalk chalk. I don't know. You know what I'm talking about, though, don't you? Yeah? go get some sidewalk chalk and with the little kids outside, draw big cells out on the street. Big ones. And then draw all the organelles. And then what you're going to do is you're going to teach those little kids about the cell. You're going to say, go step in the nucleus. And you're going to tell them about the nucleus. You're going to say, what do you find in the nucleus? And they're going to say, DNA. And then you're going to say, what does DNA stand for? And if they're at least 4 years old, they'll be able to say deoxyribonucleic acid. All right? You think I'm kidding? I'm not. I did this with my kids when they were little. All right? Yep. And I say, all right, now go to a lysosome. And they would run to a lysosome and they would say it and they'd tell me about it. So you're learning it and you're teaching them it. So when they get to college and they take their biology classes, they get it. They don't know why, but they'll say, I've got a little bit of deja vu here. I know I've done it before. All right? Now what you can do as an incentive, put some little candies. I'm not a big fan of using sweets as a reward, but put little candies in the organelles. And if they can tell you about it, they get the candy. And if they can't, then they get a good hiding. Okay?
What?
A good hiding. If it was in the US, you'd say a good whooping.
Oh.
All right? I'm just kidding. I would never advocate whooping a kid. So, kick them up the arse, that's okay. All right, so let's start by drawing our cell. Okay, there's our cell.
Don't be relatively large on our page.
Make it pretty big, but I want you to leave label space. I would make it about the length of the cell, about 2/3 the length of your page. You need label space and annotation space. All right, now again, here's what I would do. I would go out, and if you don't want to spend much money, go to the dollar store and get one of those large pieces of cardboard.
Oh yeah, they're like really sharp.
Yes, and then draw a big cell on that. And why not colour it while you're at it? Yeah? All right.
Sounds fun. I kinda want
Does. It is. Okay, good. I'll try and draw anything that is membrane. I'll try and always draw it in blue as long as I can find my blue pen. So, what's this membrane then?
What is it? A cell membrane?
It's the cell membrane. Good. Now, you already know a little bit about the structure of the cell membrane, don't you? What's a primary component of cell membrane?
Cytosol. No, that is not what it is.
Primary component of the cell membrane is?
You mean like its purpose?
Primary component. What is it? Good. Phospholipid. Right? Phospholipid. Remember those phospholipids?
Yes.
Yep.
Forms a bilayer.
Does. Okay, so there's a cell membrane. So let's start with the nucleus. Now the nucleus is probably the most obvious cell organelle. All right. Now, again, I'll elaborate on this as we talk about the nucleus, but I just want to draw it and I'll describe a little bit as I draw. So it's got a nuclear envelope, which is a double membrane. And again, I'm going to draw it kind of like this.
And I mean, the function of the envelope is that just for dual protection or something?
It's protection to contain it and to house it.
Okay.
Yeah. All right. I'm going to leave it like that, but I've got to draw. Hopefully you're drawing in pencil. I'm only going to draw a few on that side. It's got many more of them. Okay. So, I know I said the blue is membrane, and I've just drawn those in blue, so I'm actually going to go over them in black. Okay, good. All right. So, let's label this as the nucleus. Now, I'm going to stipple a dark area within the nucleus. It's not enclosed within a membrane, but it just happens to be this dense dark region. And that is the nucleolus.
Sorry, mine just looks I like it.
And then I'm going to label this as the nuclear envelope. And I'm going to label this as a pore complex. And we're going to add to this. So make sure you keep this piece of paper because we're going to continually add to it. All right? And a really good way to study this is simply to draw this a whole bunch of times with the labels and then add some annotations to your labels. All right, so the nucleus then. Let's have a look at the nucleus inside the cell. Contains most of the DNA and most of the cell's genes. There are some other places in the cell where there's DNA and there are genes, but most of them reside within the nucleus. Okay? The nucleus then is enclosed or wrapped within this nuclear envelope. And the nuclear envelope is a double layer of membrane. It's a double membrane layer. And each layer of membrane is itself a phospholipid bilayer. Okay? And the envelope itself is studded with pores. And these pores are protein complexes. And they're a little bit like a rivet in your shoe, which allows your shoelaces to go through. And that gives a physical channel for substances like proteins, which are big molecules, to move in and out, and RNA to move in and out. There's an awful lot of traffic in and out of the nucleus. There's a lot of pores, and that enables big molecules to move quickly in and out. And I'll show you some diagrams of these in a moment. Now, the nucleus does vary in size depending on the cell. But I guess you could, if you wanted a benchmark, it's about 5 microns in diameter. Okay? So a fair bit bigger than a bacterium, for example. All right. There's a lot going on in this diagram, and I'm sort of going to go over it bit by bit. So let's start up here with the cell, and there's the nucleus. Can be a fairly large part of the cell, not always, but can be. All right, so let's have a look at the nucleus then. We've got this dark region within the nucleus we call the nucleolus, and I'll elaborate on that in a moment. We've got lots of DNA. The DNA is present as a molecule called chromatin. Now, I'm not going to go into much more depth about what chromatin is. We'll do that in a later lecture. But it's present as chromatin, which is DNA, which is complexed with and wrapped around histone proteins. All right, so when we look at the nuclear envelope, we see that it's a double membrane, okay? And there are these pores made of proteins which packed at very high density. If you look at this transmission electron micrograph, there's a pore, there's a pore, there's a pore, there's a pore, there's a pore. And they're quite regular in shape, aren't they? And they're made of protein complexes. So you can imagine the open as kind of, the center is kind of being open to allow things in and out. Okay? All right. Now, when you have a look on the inside of the inner membrane, so this electron micrograph is kind of having a look at the inside of that one. Okay? There's something called the nuclear lamina. And you can see it looks net-like or mesh-like, doesn't it? It almost looks like cheesecloth. So that's a network of protein fibers that help strengthen the nucleus, give it some strength and shape. All right. So this kind of TEM, I don't know, but just looking at it, it wasn't described what it was. I think it's something called a freeze fracture TEM, or produced by something called freeze fracture. And what you do is you freeze the cell quickly in liquid nitrogen, and then you use something to cleave it, right? Like a blade, and it just breaks. But it breaks along planes of the least resistance, all right? So this kind of looks a little bit three-dimensional. So, but you can see the surface then of the nucleus. There you've got the outer membrane, and you can see where the inner membrane is sort of underneath it because the outer membrane broke off and the other part was sort of fractured away. And you can also see these pore complexes. All right. Is everybody okay then with nucleus? Yep. All right. Okay, so the nucleolus then. Sometimes on an electron micrograph, this dark line is the nuclear envelope, and here's the nucleolus. It's a very darkly stained region. And if we were to diagram it, again, it is darkly stained region. Is the nucleolus itself wrapped within a membrane? No, it's not. All right. So the nucleolus then is this dark or dense region in the nucleus. And it's the region in the nucleus where ribosomal RNA is made. Now, we have talked about RNA, but we've not talked about the different kinds of RNA. Ribosomal RNA, as the name implies, are what ribosomes are made up of. All right? And we'll talk about ribosomes next. So it's where ribosomal RNA is produced, and then we take the RNA and we complex it with some proteins, and then we can assemble ribosomal subunits. Not yet?
Not yet.
Sorry. That was enthusiasm for you, right? I don't want to know what book they're reading now.
I know.
Huh?
We bursted in and I was like, why Britain's so heavy?
All right. Yeah, you could have joined, right? Sorry. Okay. All right. Everybody good about the nucleolus then? It's the site where ribosomes are essentially made. The RNA is complexed with proteins to form these ribosomal subunits. All right. So let's look at ribosomes then. Okay, ribosomes. So let's draw some ribosomes on our cell first. All right, so we've got to add some more structures. So do you notice how I've drawn these? That outer membrane of the nuclear envelope is sort of an extension now into this. Is everybody okay with that? Now in a different colour, I'm going to draw some dots, and it's these little dots that are the ribosomes. So I'm going to draw a whole bunch of the ribosomes here, here. And I'm not going to cover this whole thing because some areas don't have ribosomes and some areas of it have many ribosomes. But I'm only going to do it on this one half. Now here's what I want you to see is that we've got now a separation. There's an inside of this structure, which is called the endoplasmic reticulum, and then we've got the outside. Okay? Now I'm going to draw a bunch of these dots in the cytoplasm on the outside.
Are they also ribosomes?
If it's a red dot, it's a
Yeah,
ribosome.
that's what I was-
Yep, and I'll label that. Okay? All right, so first I'm going to label this structure I just drew. Okay? So this is called the smooth. Now right now I'm going to call it the smooth ER, and I'm going to call this region of it the rough ER. And together these are two regions of the endoplasmic reticulum. All right, the endoplasmic reticulum. red dots that I've drawn are ribosomes. The ones that are attached, kind of embedded but attached to the ER, they're bound ribosomes. Bound ribosomes. And the ones that are just sort of floating in the cytoplasm outside of the ER and unattached are called free ribosomes. Okay, good so far? So ribosomes then, they are enormous molecular complexes. You can see them on transmission electron micrographs of the cell very clearly as these little granules. They're enormous molecular complexes made of ribosomal RNA and proteins. Now, the ribosomes that we have as eukaryotes, we can refer to them by their size, and we call them ATS ribosomes. Now, just call them ribosomes, but eukaryotes have something called ATS ribosomes. Now, the S is a measure of how quickly they precipitate out in a centrifuge when you centrifuge them. All right? It's sort of a measure of how easily they are to pellet out in a centrifuge. And we've done centrifugation, haven't we? Yep. And you know that some things precipitate easily and quickly, some things not. Ribosomes take a long time. You've got to spin it fast and for a long time. But ATS is a measure. And the ATS ribosome itself is made of two subunits, a 40S and a 60S subunit. There's a small subunit, which is a 40S, and a large subunit, which is a 60S. Okay? So when the large and the small subunits come together, we end up with an ATS ribosome. Now, it's important to know that because bacteria have a different kind of ribosome. Now, it's very similar, but it's a little bit smaller. It's a 70S ribosome. It's also made of two subunits, but its overall size is a little bit smaller. Okay? All right. Now, it's important to know the difference between bacterial and eukaryotic ribosomes because certain antibiotics that you take specifically hit 70S ribosomes, not ATS ribosomes. And it's one reason why certain antibiotics affect bacteria and not you. If you take out the ribosome, what are you going to stop the cell from doing? Making proteins. That's instant disaster, death for the cell. Take out the 70S ribosome and you only kill bacteria. Okay. All right. So, ribosomes carry out protein synthesis. They are the site where amino acids are joined together to form polypeptide chains. And there are two places that you're going to find ribosomes. In the cytosol, which are the free ribosomes, they're just sort of like floating in this gelatinous matrix, not attached to anything. Yep.
Sorry, what did you say about polypeptides and did you say photosynthesis? Is that what you said?
It's the ribosomes are the site where amino acid monomers are joined to form polypeptide chains. And again, we'll look at that in a lot more depth in a later chapter when we look at how proteins are made from DNA. So the other place you're going to find them is on the outside of the endoplasmic reticulum. we call those the bound ribosomes. Now you can also find ribosomes on the outer membrane of the nuclear envelope. But a bound ribosome is one that's attached to a membrane.
So it happens in two different areas because there's two different locations of ribosomes.
Two different places you can find them. They're no different to each other. The free ones and the bound ones are the same. They're just ribosomes. But sometimes they're floating in the cytoplasm, sometimes they become attached and they're bound ones.
And they're not necessarily floating next to the smooth or the rough or the, I guess, the area. They can be in different parts.
They can be out. They tend to be located around where the ER is. Yeah, they can be another place in the cell. Yeah.
All right.
All right. So is everybody okay with the smooth ER? Everybody's okay with the rough ER?
So the smooth is just towards the outside of it?
The smooth is because it has no ribosomes bound to it. We'll talk about the ER separately in just a sec. All right. Bound and free ribosomes. You're good with those, yeah? All right. So this is a computer-generated model of what a ribosome looks like. All right. And they are enormous molecules, proteins and RNA complex together. Here's the large ribosomal subunit, and there's the small ribosomal subunit. Okay. So sometimes we draw them, rather than drawing this, that would be a handful to draw, wouldn't it? Yeah? Well, let's just draw them like that. 2 parts. Easy. Here we've got the large subunit stacked on top of the small subunit. And they can be free of one another, or they can come together and join one another. Okay? And if you were to have a look, a close-up of this area here, then the TEM would look a little bit like this. Here's the endoplasmic reticulum. Those granules that are attached to the ER membrane, they're bound ribosomes, and these ones that are just not attached, they're the free ribosomes. Okay? All right. Good? Okay. So this diagram kind of puts it all in perspective. There's your endoplasmic reticulum, which is sort of contiguous with the outer envelope of the nucleus, what a TM would look like, and you've got your free and bound ribosomes, and that's how we can draw a ribosome. Oh. Actually, that slide was a mistake. I should have removed it. I'll remove it when I post it. All right, so let's see, how are we doing for time? I think we'll wrap it up there on the endoplasmic reticulum. Right, so what are you going to go home and practice? Drawing the cell. You've got 1, 2, 3, 4, 5, 6, 7, 8, 9 labels to learn and draw. Easy. And their functions. Easy. Draw this 2, 3 or 4 times, and if you goof around with a little kid, you've got it. Locked in, you won't forget it.